5.6.2 tRNA, the adapter molecule. From the very beginning of the proposition of code, it was clear to Francis Crick that there has to be a mechanism to read the code and also to link it to the amino acids. Because amino acids have no structural specialities to read the code uniquely. He postulated the presence of an adapter molecule that would on one hand read the code and on other hand would bind to specific amino acids. The tRNA, then called sRNA, soluble RNA, was known before the genetic code was postulated. However, its role as an adapter molecule was assigned much later. tRNA has an anticodon loop that has bases complementary to the code, and it also has an amino acid acceptor end to which it binds to the amino acids. tRNA are specific for each amino acid, figure 5.12. For the initiation, there is another specific tRNA that is referred to as initiator tRNA. There are no tRNAs for stop codons. In figure 5.12, the secondary structure of tRNA has been depicted that looks like a clover leaf. In actual structure, the tRNA is compact molecule which looks like inverted L. 5.7 Translation. Translation refers to the process of polymerization of amino acids to form a polypeptide, figure 5.13. The order and sequence of amino acids are defined by the sequence of bases in the mRNA. The amino acids are joined by a bond which is known as a peptide bond. Formation of peptide bond requires energy. Therefore, in the first phase itself, amino acids are activated in the presence of ATP and linked to their cognate tRNA, a process commonly called as charging of tRNA or amino acylation of tRNA to be more specific. If two such charged tRNAs are brought close enough, the formation of peptide bond between them would be favored energetically. The presence of a catalyst would enhance the rate of peptide bond formation. The cellular factory responsible for synthesizing proteins is the ribosome. The ribosome consists of structural RNAs and about 80 different proteins. In its inactive state, it exists as two subunits, a large subunit and a small subunit. When the small subunit encounters the mRNA, the process of translation of the mRNA to protein begins. There are two sites in the large subunit for subsequent amino acids to bind to and thus be close enough to each other for the formation of a peptide bond. The ribosome also acts as a catalyst, that is 23S rRNA in bacteria is the enzyme ribozyme for the formation of peptide bond. A translation unit in mRNA is the sequence of RNA that is flanked by the start codon AUG and the stop codon and codes for a polypeptide. An mRNA also has some additional sequences that are not translated and are referred to as untranslated regions UTR. The UTRs are present at both 5' end before start codon and at 3' end after stop codon. They are required for efficient translation process. For initiation, the ribosome binds to the mRNA at the start codon AUG that is recognized only by the initiator tRNA. The ribosome proceeds to the elongation phase of protein synthesis. During this stage, complexes composed of an amino acid linked to tRNA sequentially bind to the appropriate codon in mRNA by forming complementary base pairs with the tRNA anticodon. The ribosome moves from codon to codon along the mRNA. Amino acids are added one by one, translated into polypeptide sequences dictated by DNA and represented by mRNA. At the end, a release factor binds to the stop codon, terminating translation and releasing the complete polypeptide from the ribosome. 5.8 Regulation of gene expression. Regulation of gene expression refers to a very broad term that may occur at various levels. Considering that gene expression results in the formation of a polypeptide, it can be regulated at several levels. In eukaryotes, the regulation could be exerted at number 1 transcriptional level, that is formation of primary transcript, Number 2, processing level, that is regulation of splicing. Number 3, transport of mRNA from nucleus to the cytoplasm. And the last, number 4, translational level. The genes in a cell are expressed to perform a particular function or a set of functions. For example, if an enzyme called beta galactosidase is synthesized by E. coli, it is used to catalyze the hydrolysis of a disaccharide, lactose, into galactose and glucose. The bacteria use them as a source of energy. Hence, if the bacteria do not have lactose around them to be utilized for energy source, they would no longer require the synthesis of enzyme beta galactosidase. Therefore, in simple terms, it is the metabolic, physiological or environmental conditions that regulate the expression of genes. The development and differentiation of embryo into adult organism are also a result of coordinated regulation of expression of several sets of genes. In prokaryotes, control of the rate of transcriptional initiation is the predominant site for control of gene expression. In a transcription unit, the activity of RNA polymerase at a given promoter is in turn regulated by interaction with accessory proteins, which affects its ability to recognize start sites. These regulatory proteins can act both positively, that is activators, and negatively, that is repressors. The accessibility of promoter regions of prokaryotic DNA is in many cases regulated by the interaction of proteins with sequences termed operators. The operator region is adjacent to the promoter element in most operons, and in most cases the sequences of the operator bind a repressor protein. Each operon has its specific operator and specific repressor. For example, lac operator is present only in the lac operon and it interacts specifically with lac repressor only. 5.8.1 The lac operon. The elucidation of the lac operon was also a result of a close association between a geneticist, Francois Jacob, and a biochemist, Jacques Monod. They were the first to elucidate a transcriptionally regulated system. In lacoperon, here lac refers to lactose, a polycistronic structural gene is regulated by a common promoter and regulatory genes. Such arrangement is very common in bacteria and is referred to as operon. To name a few such examples, lac operon, TRP operon, era operon, his operon, val operon, etc. The lac operon consists of one regulatory gene, the I gene. Here the term I does not refer to inducer, rather it is derived from the word inhibitor, and three structural genes Z, Y and A. The I gene codes for the repressor of the lac operon. The Z gene codes for beta galactosidase beta gal, which is primarily responsible for the hydrolysis of the disaccharide lactose into monomeric units galactose and glucose. The Y gene codes for permease, which increase permeability of the cell to beta galactosides. The A gene encodes a trans-acetylase, hence all the three gene products in lac operon are required for metabolism of lactose. In most other operons as well, the genes present in the operon are needed together to function in the same or related metabolic pathway. Figure 5.14. Lactose is the substrate for the enzyme beta galactosidase and it regulates switching on and off of the operon. Hence, it is termed as inducer. In the absence of a preferred carbon source such as glucose, if lactose is provided in growth medium of the bacteria, the lactose is transported into the cells through the action of permease. Remember, a very low level of expression of lac operon has to be present in the cell at the time. Otherwise, lactose cannot enter the cells. The lactose then induces the operon in the following manner. The repressor of the operon is synthesized all the time constitutively from the I gene. The repressor protein binds to the operator region of the operon and prevents RNA polymerase from transcribing the operon. In the presence of an inducer such as lactose or allolactose, the repressor is inactivated by interaction with the inducer. This allows RNA polymerase access to the promoter and transcription proceeds. Figure 5.14. Essentially, regulation of lac operon can also be visualized as regulation of enzyme synthesis by its substrate. Remember, a glucose or galactose cannot act as inducers for lac operon. Can you think for how long the lac operon would be expressed in the presence of lactose? Regulation of lac operon by repressor is referred to as a negative regulation. Lac operon is under control of positive regulation as well, but it is beyond the scope of discussion at this level. 5.9 Human Genome Project. In the preceding section, you have learned that it is the sequence of bases in the DNA that determines the genetic information of a given organism. In other words, genetic makeup of an organism or an individual lies in the DNA sequences. If two individuals differ, then their DNA sequences should also be different, at least at some places. These assumptions lead to the quest of finding out the complete DNA sequence of human genome. With the establishment of genetic engineering techniques where it was possible to isolate and clone any piece of DNA and availability of simple and fast techniques for determining DNA sequences, a very ambitious project of sequencing human genome was launched in the year 1990. Human Genome Project HGP was called a mega project. You can imagine the magnitude and the requirements for the project if we simply define the aims of the project as follows. Human genome is said to have approximately 3 into 10 to the power 9 base pair, and if the cost of sequencing required is US dollar 3 per base pair, the estimated cost in the beginning, the total estimated cost of the project would be approximately 9 billion US dollars. Further, if the obtained sequences were to be stored in typed form in books, and if each page of the book contained 1,000 letters and each book contained 1,000 pages, then 3,300 such books would be required to store the information of DNA sequence from a single human cell. The enormous amount of data expected to be generated also necessitated the use of high-speed computational devices for data storage and retrieval and analysis. HGP was closely associated with the rapid development of a new area in biology called bioinformatics. Goals of HGP. Some of the important goals of HGP were as follows. Number 1, identify all the approximately 20,000 to 25,000 genes in human DNA. Number 2, determine the sequences of the 3 billion chemical base pairs that make up human DNA. Number 3, store this information in databases. Number 4, improve tools for data analysis. Number 5, transfer related technologies to other sectors such as industries. Number 6, and the last one, address the ethical, legal, and social issues LCELSI that may arise from the project. The Human Genome Project was a 13-year project coordinated by the US Department of Energy and the National Institute of Health. During the early years of HGP, the Wellcome Trust UK became a major partner. Additional contribution came from Japan, France, Germany, China, and others. The project was completed in 2003. Knowledge about the effects of DNA variations among individuals can lead to revolutionary new ways to diagnose, treat, and someday prevent the thousands of disorders that affect human beings. Besides providing clues to understanding human biology, learning about non human organisms' DNA sequences can lead to an understanding of their natural capabilities that can be applied towards solving challenges in healthcare, agriculture, energy production, environmental remediation. Many non human model organisms, such as bacteria, yeast, Caenorhabditis elegans, a free-living non-pathogenic nematode, Drosophila, the fruit fly, plants, rice and Arabidopsis, etc. have also been sequenced. Methodologies. The methods involved two major approaches. One approach focused on identifying all the genes that are expressed as RNA, referred to as expressed sequence tags (ESTS). The other took the blind approach of simply sequencing the whole set of genome that contained all the coding and non-coding sequence and later assigning different region in the sequence with functions, a term referred to as sequence annotation. For sequencing, the total DNA from a cell is isolated and converted into random fragments of relatively smaller sizes. Recall DNA is a very long polymer and there are technical limitations in sequencing very long piece of DNA and cloned in suitable host using specialized vectors. The cloning resulted into amplification of each piece of DNA fragment so that it subsequently could be sequenced with ease. The commonly used hosts were bacteria and yeast, and the vectors were called as BAC, bacterial artificial chromosome, and YAC, yeast artificial chromosomes. The fragments were sequenced using automated DNA sequencers that worked on the principle of a method developed by Frederick Sanger. Remember, Sanger is also credited for developing method for determination of amino acid sequences in proteins. These sequences were then arranged based on some overlapping regions present in them. This required a generation of overlapping fragments for sequencing. Alignment of these sequences was humanly not possible. Therefore, specialized computer-based programs were developed. Figure 5.15. These sequences were subsequently annotated and were assigned to each chromosome. The sequence of chromosome 1 was completely only in May 2006. This was the last of the 24 human chromosome, 22 autosome and X and Y to be sequenced. Another challenging task was assigning the genetic code and physical maps on the genome. This was generated using information of polymorphism of restriction endonuclease recognition sites and some repetitive DNA sequences known as microsatellites. One of the applications of polymorphism in repetitive DNA sequence shall be discussed in the next section of DNA fingerprinting. 5.9.1 Salient features of human genome. Some of the salient observations drawn from Human Genome Project are as follows. Number 1. The human genome contains 3164.7 million base pair. Number 2. The average gene consists of 3000 bases but sizes vary greatly, with the largest human known gene being dystrophin at 2.4 million bases. Number 3, the total number of genes is estimated at 30,000, much lower than previous estimates of 80,000 to 1,40,000 genes. Almost all 99.9% nucleotides bases are exactly the same in all people. Number 4, the functions are unknown for over 50% of the discovered genes. Number 5. Less than 2% of the genome codes for proteins. Number 6. Repeated sequences make up very large portion of the human genome. Number 7. Repetitive sequences are stretches of DNA sequences that are repeated many times, sometimes 100 to 1000 times. They are thought to have no direct coding functions, but they shed light on chromosome structure, dynamics, and evolution. Number 8. Chromosome 1 has the most genes, 2968, and the Y has the fewest, that is, it has only 231. Number 9. Scientists have identified about 1.4 million locations where single base DNA differences (SNPs), single nucleotide polymorphism pronounced as SNPs, occur in humans. This information promises to revolutionize the process of finding chromosomal locations for disease associated sequences and tracing human history. 5.9.2 Applications and future challenges. Deriving meaningful knowledge from the DNA sequences will define research through the coming decades, leading to our understanding of biological systems. This enormous task will require the expertise and creativity of tens of thousands of scientists from varied disciplines in both the public and private sectors worldwide. One of the greatest impacts of having the HG sequence may well be enabling a radically new approach to biological research. In the past, researchers studied one or few genes at a time. With whole genome sequences and new high throughput technologies, we can approach questions systematically and on a much broader scale. They can study all the genes in a genome, for example, all the transcripts in a particular tissue or organ or tumor, or how tens of thousands of genes and proteins work together in interconnected networks to orchestrate the chemistry of life. 5.10 DNA fingerprinting. As stated in the preceding section, 99.9% of base sequence among humans is the same. Assuming human genome as 3 into 10 to the power 9 base pair, in how many base sequences would there be differences? It is these difference in sequences of DNA which make every individual unique in their phenotypic appearance. If one aims to find out genetic differences between two individuals or among individuals of a population, sequencing the DNA every time would be a daunting and expensive task. Imagine trying to compare two sets of 3 into 10 to the power 6 base pairs. DNA fingerprinting is a very quick way to compare the DNA sequences of any two individuals. DNA fingerprinting involves identifying differences in some specific regions in DNA sequence called as repetitive DNA, because in these sequences a small stretch of DNA is repeated many times. These repetitive DNA are separated from bulk genomic DNA as different peaks during density gradient centrifugation. The bulk DNA forms a major peak and the other small peaks are referred to as satellite DNA. Depending on base composition, AT rich or GC rich, length of segment and number of repetitive units, the satellite DNA is classified into many categories such as microsatellites and mini satellites, etc. These sequences normally do not code for any proteins, but they form a large portion of human genome. These sequences show high degree of polymorphism and form the basis of DNA fingerprinting. Since DNA from every tissue such as blood, hair follicle, skin, bone, saliva, sperm, etc. from an individual show the same degree of polymorphism, they become very useful identification tool in forensic applications. Further, as the polymorphisms are inheritable from parents to children, DNA fingerprinting is the basis of paternity testing in case of disputes. As polymorphism in DNA sequence is the basis of genetic mapping of a human genome as well as of DNA fingerprinting, it is essential that we understand what DNA polymorphism means in simple terms. Polymorphism, that is variation at a genetic level, arises due to mutation. Recall different kind of mutation and their effects that you have already studied in chapter 4 and in the preceding section in this chapter. New mutations may arise in an individual either in somatic cells or in germ cells, cells that generate gametes in sexually reproducing organism. If a germ cell mutation does not seriously impair individual's ability to have offspring who can transmit the mutation, it can spread to the other members of population through sexual reproduction. Allelic, again recall the definition of alleles from chapter 4. Allelic sequence variation has traditionally been described as a DNA polymorphism if more than one variant, that is allele, at a locus occurs in human population with a frequency greater than 0.01. In simple terms, if an inheritable mutation is observed in a population at a high frequency, it is referred to as a DNA polymorphism. The probability of such variation to be observed in non-coding DNA sequence would be higher as mutation in these sequences may not have any immediate effect or impact in an individual's reproductive ability. These mutations keep on accumulating generation after generation and from one of the bases of variability or polymorphism. There is a variety of different types of polymorphism ranging from single nucleotide change to very large scale changes. For evolution and speciation, such polymorphism play very important role and you will study these in details at higher classes. The technique of DNA fingerprinting was initially developed by Alec Jeffreys. He used a satellite DNA as a probe that shows very high degree of polymorphism. It was called as variable number of tandem repeats VNTR. The technique as used earlier involved southern blot hybridization using radio labeled VNTR as a probe. It included number 1 isolation of DNA, number 2 digestion of DNA by restriction endonucleases, number 3 separation of DNA fragments by electrophoresis, number 4 transferring blotting of separated DNA fragments to synthetic membranes such as nitrocellulose or nylon, number 5 hybridization using labeled VNTR probe, and number 6 detection of hybridization DNA fragments by autoradiography. A schematic representation of DNA fingerprinting is shown in figure 5.16. The VNTR belongs to a class of satellite DNA referred to as mini satellite. A small DNA sequence is arranged tandemly in many copy numbers. The copy numbers varies from chromosome to chromosome in an individual. The numbers of repeat show very high degree of polymorphism. As a result, the size of VNTR varies in size from 0.1 to 20 kb. Consequently, after hybridization with VNTR probe, the autoradiogram gives many bands of differing sizes. These bands give a characteristic pattern for an individual DNA, figure 5.16. It differs from individual to individual in a population except in the case of monozygotic, that is identical twins. The sensitivity of the technique has been increased by use of polymerase chain reaction PCR. You will study about it in chapter 9. Consequently, DNA from a single cell is enough to perform DNA fingerprinting analysis. In addition to application in forensic science, it has much wider applications such as in determining population and genetic diversities. Currently, many different probes are used to generate DNA fingerprints.